This chapter talks about sensation and perception. If you're interested in some of the material that we discuss here, you can take an entire semester on sensation and perception. And some of the concepts that we talk about, they will go in depth into it. For our class, fortunately, unfortunately, we are going to touch very, very broadly on some of these concepts. So we're going to start off with three important definitions. Please make sure you know all three for the exam. Sensation simply is the detection of external stimuli and the transmission of this information to the brain. So sensation is picking up information through any of your five senses and sending that information into the brain. Perception is how we process, organize, and interpret these sensory signals. So the message comes in, it goes into the brain, and the brain makes sense of it, organize it, and interprets these sensory signals through a process called perception. Think of transduction as translation. This is taking the stimuli and coding it into, translating it into a language that the brain understands, which are these electric signals. So as we go through each sense, this will make a bit more sense. Nothing here you need to know, but sub meaning under, subliminal stimuli. These are stimuli that may be all around us, but we're not consciously aware of it, meaning it ignites or triggers the senses, but it's not enough for us to be aware of it. There may be sounds around us enough where we don't pick up. And maybe your dog starts barking and you have no idea what they're barking at because they can hear it, but it's subliminal stimuli for us. So just below the threshold of our consciousness. Again, these two you don't need to know. If you take the class, they will talk extensively about these. Absolute threshold is the smallest amount of energy needed for a person to consciously detect the stimuli 50% of the time that's present. So for example, they want to know what is your absolute threshold for hearing stimuli. So they will play sounds, or they will have sounds, and they will increase the sound until you can detect the sound at least 50% of the time. And that would be the absolute threshold for your auditory sense. Just noticeable difference, or JND, the difference threshold is at what point would you know when one sound is louder than another sound. So this is the smallest difference between two stimuli that the individual can detect, again, 50% of the time. So we're going to go through 5 senses. Some of them will have a technical term associated with it. You got to know that for the exam. So we're going to start off with a sense of touch, and the technical term for that is haptic. Sense of touch has these 3 characteristics. So when you touch something, you might notice the temperature of it, the pressure, and the pain, if pain is associated. So this ice bucket challenge that was done a while ago really demonstrated these 3 different characteristics of sense. So as the ice and water is going over a person's head, they feel the temperature, which is cold, and it's cold enough for them to feel the pain of it, and the pressure as the water goes through. So the really noticing all 3 characteristics. Kinesthetic sense is the perception of the position in space and movement of our bodies and our limbs. Dancers have a really good kinesthetic sense. Gymnasts have a really good kinesthetic sense, the perception of their bodies according to space and their limbs. So just the terminology. You don't need to know any of this for the exam, but we know sensory receptors within the skin fire when the surface is touched, And what happens is, let's say you want to close your eyes and someone gives you something and says, guess what it is? Well, you wouldn't just hold it in your hand, you would kind of go around it. This is what we call active touching, which is continuously moving one's hand along the surface of an object so that we can perceive, or our brain can give us a complete perception of the object. We know some of the areas of the bodies are more sensitive than others. For example, getting hit on the nose for almost everyone is painful, but there is individual differences in what areas of the body are more sensitive than others. Some people are really sensitive around their neck, and that may be because there's more nerve ending densely packed in that area versus other folks. Again, this you don't need to know for the exam. Receptors are located right beneath the skin. So when the skin temperature increases, receptors for warmth will fire. When they decrease, one will perceive cold. And sensation for temperature is really relative. What's fascinating about our perception of temperature is that when you're younger, and I mean by younger, 20s, I mean obviously children, but 20s, 30s, we'll say even maybe up to 40s, our body has a way of adjusting to our environment. So let's say you have lived in Alaska for years and years and years, and you come here, and for us it would be a cold day. You can see these folks in really shorts and short sleeve because their perception of temperature is different. And the longer they're here, then they will begin to feel the cold like you and I would. Except, with the exception, as you get older, your ability to deal with the cold is a bit more difficult. You become more sensitive to the cold. So that's one of the reasons folks move to the south quite often, a southern part of the country, is because of that, retiring in Florida or such. This you need to know for the exam. So before the opioid epidemic in the US, the doctors believed that taking pain medication was highly addictive, and we're not crazy about giving out these pain medication, these opioids. So there were clinics, pain control clinics, that were trying to use more psychological and non-medical ways of helping folks deal with chronic pain. So we had all these theories and these therapeutic techniques that came out. One of the most credible one was this pain gate control theory that says pain, we know pain involves biological, psychological, and cultural factors. If you pay attention to the pain, you will feel it more than if you try to distract yourself. One of the reasons is when pain receptors are activated somewhere in the body, it gets picked up and sent to the spinal cord. The spinal cord then sends it to the brain, to the pain perceiving area of the brain, and the person will begin to feel it. So people who unfortunately are paralyzed, let's say from waist down, if they get hurt or break a leg, they will not feel it. Part of it is those receptors are unable to send that message through the spinal cord because there's a break into the brain. So that will never reach the brain, thus they don't feel the pain. So this theory said when you do feel pain, there is a gate that opens in the spinal cord that allows a signal to be carried by nerve fibers. So if you can compete for that gate, instead of just one sensation going through the gate, maybe you have two. So when you get hurt, and parents quite often do this, when their child hits their head somewhere and the pain is beginning, or their leg, let's say, the pain starts going to the brain, the child starts crying. Quite often we rub the area or kiss it. This is a competing stimuli, so the rubbing the pain area will compete for neural attention, therefore decreasing the perception of pain. So this is a therapeutic technique that quite often works for folks. Your second sense is a sense of vision, which is really important. There's been a lot of studies done for humans because we are so visually dependent. We depend on vision for um, perceiving danger, for interacting, for just everyday life. And what they have found is we have these really wavelengths all around us, but human brain can only detect a very small portion of within the spectrum. So this is apparently what we can see in terms of these waves and these colors. So this you don't need to know for the exam, but the brain doesn't see the world in photograph. It needs to transduce the information into electric charge that the brain understands or electric signals that the brain understands. Everything on this page you need to know, and I apologize, I didn't put it on the PowerPoint, but you need to know the different areas of the eye, what their function is. You don't need to know the location, but you do need to know the function. So we're going to start off with outside area and then work our way to inside of the brain, the eye. Right here, if you ever get a chance, if you have someone that can volunteer and you can kind of take a look at their eye, look at the periphery, have them face forward and you stand on the side and have them open their eyes and you can see some of the outside portion. The cornea, which is this, it should be nice and round, is a clear jelly-like structure that helps bend light rays. So cornea is the clear covering of the eye. The iris is the color portion, and we know each of our irises are very individualized to us. And make sure you know this for the exam or write it down somewhere. If you've seen in the movies, they have fingerprints, right? They take a fingerprint of someone to identify them. In the futuristic films, they're actually looking at their eyeballs. If you've seen them, they scan the eyeball. Well, what we know is your iris, the pattern portion of your iris, the color portion, we have some specks in others and lines. Your iris is more individualized to you than even your fingerprints. So it's a very individualistic pattern, has its own pattern. The pupil is the opening that lets light and other stimuli enter the eye. The pupil, it looks like it's black. Part of it is just that hole. And the pupil, depending on the amount of light that's out, will expand or constrict. And it does it through these, dependent on light. Sclera is the white portion of the eye. Ciliary muscles, they help the lens adjust its thickness so that we can see far or close. And ultimately, the lens will project the vision to the back of the eyes for processing. When your lens doesn't, and they got the idea of camera basically from here. When your lens projects the vision to the right area of the eye, then you have 20/20 vision. When the lens doesn't do what it's supposed to, then we have those far-sighted or nearsighted issues. So the lens will project to the back of the eye. We're going to talk about the retina and specifically the fovea. You got to know this for the exam, so we'll talk about these two in a second. If you look right here, this is the yellow. This is filled with photoreceptor nerves. And what they do is they pick up the information. This gives us peripheral vision. It picks up the information through the optic nerve, sends it to the eye for processing. But there's a small area in the eye where there is no vision. There are no nerves. So this is what we call the blind spot. There will be no vision in this area of the eye. So if you remember, we talked about that area in the back of the eye called fovea. Fovea, and make sure you write this down, fovea provides the sharpest, most detailed image for you. And part of the reason, it is rich in cones. So those photoreceptors that we have in the back of the eye, we have cones and rods provide us with different forms of information, translate information differently. Cones enable you to see color. So people that are colorblind, their cone for certain color may not be working, at least that's the theory. So cones enable you to see color and really detail. Rods transduce information about black and white and are found usually in the periphery of retina. They also work at night when you don't have a lot of light, you can't see color, that's your rods working. Make sure you know all three of them actually for the exam. So what happens here is these light rays come in, and if you can see the cornea, what the cornea is able to do is bend it. So when it comes here, it bends and sends it through the pupil. The lens will help you focus and send it to the right area of the eye for processing. Now this yellow area, if you look, we have the cones and rods that enable you to see color and black and white and the peripheral. And then they will send it through these cells to the optic nerve. Optic nerve then will pick the information, transduce it into an electrical signal, and through the optic nerve send it to the brain. Everything on here, please make sure you know, and it's just exactly what I just said. So light is emitted or reflected into the eye, and it enters through the pupil, that black opening in the center of the eye, passing through the lens, which enables it, projects it to the right area, back of the eye. The light excites the photoreceptors on the retina, the back of the eye. Photoreceptors will then transduce or translate the information into an electric signal that the brain understands. And what happens is it goes through the optic nerve, ultimately to the visual cortex of the occipital lobe, which is in the back of your basic back of your head. Optic nerve crosses over at this center. Think of it like Grand Central Station for vision, called optic chiasm. I'm going to show you what that looks like. And what we know is the left visual field is processed by the right hemisphere and vice versa. So your eye is picking up information. This is your optic nerve. They converge at a point called optic chiasm. They come together at this point, and what's coming from the left will then cross over and go over to this area, lateral geniculate nucleus. This is actually a small area within the thalamus. If you remember from chapter 2, we talked about thalamus being the portion or the area of your sensory, oh my God, I'm blanking, your sensory translator, right? So all the senses go through the thalamus except for the sense of smell, and then it gets sent to the right area of the brain for processing. So within the thalamus, there's different areas for each of our senses. The lateral geniculate nucleus, a cluster within the thalamus, is responsible for vision. So as information comes from the left nerve, crosses over and goes to the right lateral geniculate nucleus, and then will be processed by the primary visual cortex, so you know what you're looking at. So this is what we're talking about here. But you know, because we have peripheral vision, left visual field may pick up information also from the right visual field. So it's not like we're only looking at, they will cross over. But whatever information comes from here goes to the optic chiasm, then will go into the, so if you can see the blue, goes over to the lateral geniculate nucleus, which is your area in the thalamus, and then will be processed by the right occipital lobe area. Please make sure you know the technical term for nearsighted or farsighted. Nearsightedness or myopia is when the focal point falls short of the retina, so the individual is unable to see far, myopia. Farsightedness is the inability to really see close, so you can't see far. The focus is right behind the retina, and this is called hyperopia. So make sure you know those are both of these for the exam. So you might see, what's another term for nearsighted? So And this is what basically is happening. So the lens is not able to do its job, is not able to project it to the focal point, unfortunately, and we get visual disturbances. We as humans are pretty good at perceiving and recognizing faces. You don't need to know any of these, but our perception of faces and what they mean in terms of their emotion will differ. What you do need to know is prosopagnosia. This is a condition in certain individuals that they have a deficit in the ability to recognize faces. So if they've seen a face before, it is difficult for them to, again, to recognize that person, let's say second or third time. Prosopagnosia. There's a lot of theories about color vision. One of the most popular one is this trichromatic theory. Please make sure you know this for the exam. This is says our ability to process color is because of these three cones that we have, and a combination of all of them will give us the full color spectrum. We have red, blue, and green cones that enable us to see color. And when you're colorblind for one of them, let's say green, you're not able to see shades of green and some of the colors that are associated with the green tone. So in color blindness, you have monochrome color blindness. These individuals, cones isn't working at all or is dysfunctional. They either have no cones or it's not working. Therefore, the person sees in black and white, which is mono one chrome color blindness. You have red or green. These are individuals whose red or green cones may not be working. Again, those are more specific color vision problems. So this is a test that they use for color blindness test. And there's numbers in the middle. I'm going to give you a minute to look at the numbers. If you don't see the numbers, you might want to talk to your optometrist. So here you should be able to see the color 7, 13, 16, 8, 12, and this is 9. Again, if you can't see any of the numbers, you might want to check it out. We have this really interesting perceptual distortion called after image, which is a lingering visual impression made by a stimulus that has been removed. So I'm going to show you a vision. If you want to participate in this, after you see the image, pause the slide and stare at it at about 30 seconds. You can time it. So you can get a timer ready. Once I click, stare at the image and then look away and see if the image continues to exist. This is what we call after image. So stare at the center, and you might want to put this on pause. After 30 seconds, look away and see if you still have, you see the stars or the stripes. That's after image. Our third sense is the auditory sense. If you've ever heard people say in space, no one can hear you scream, one of the reasons is that there is no medium for sound to travel in space. On Earth, we have sound requires a medium, and it's air that travels. If you go underwater and scream, it's more muffled because it's more dense, so you can't hear it as well. But air is optimal for sound to travel. Sound is transmitted through compression of air traveling in waves, creating these vibrations. For the exam, there's different qualities to the brain processing sound. The ones you need to know for the exam is pitch and volume. The pitch is the sound's frequency or number of cycles per second. It is measured in this measurement called hertz. Please make sure you know that for the exam. Timbre is the quality or character of a sound, the tone of it. Volume is the loudness or the amplitude of sound waves, and it is measured in decibels. So make sure you know that for the exam. Then we have location of sound. When we hear something, our brain is able to process, so hopefully we can turn to the location of the sound. None of these you need to know for the exam, but we know there's a 60-60 rule, although with these new headphones coming on, I don't know how many people are really going by this rule, but the rule is you shouldn't listen to more than 60% of the full volume for more than 60 minutes per day. If you do, you are unfortunately may be causing hearing loss. Those individuals who work at the airport without wearing those big old headphones, even with wearing it, there is evidence of hearing loss. iPod oblivion, when we're listening, because our brain is processing that information, we may be a little bit less aware of our surroundings. So we know when folks are talking on the phone or listening to music and singing away, they're not as aware of their surroundings when driving as no sound at all. The ear, ultimately, that's how sound comes in through vibration. This is the three different parts, the outer ear, middle ear, and inner ear. I am not going to go through this, and you don't need to know any of these. I want you to kind of get an idea. Those of you that have taken anatomy, apparently the words that they use is a little bit different than ours. But what I want you to pay attention to is this cochlea and the vestibular nerve, or the cochlear nerve. So in the outer ear funnels sound to the eardrum. Middle ear, you have the eardrum, hammer, anvil, and stirrup. And again, the words that used is a little bit different, but make sure you know the middle ear acts as an amplifier. So when the middle ear, unfortunately, is damaged through loud sound or even physical hits, it can damage the amplifier and people might lose their hearing. The inner ear is the most sensitive, I would say. You have this auditory nerve. So make sure you know this, no, not this one, I'll show you this. These are two different types of deafness, and there are different techniques not techniques, but treatments to help those. Please make sure you know both of them, the cause and ultimately how to treat it. Conductive deafness is that amplifier, right? Somehow that amplifier is damaged, not quite often seen as you age with a lot of sound. You might lose this ability to really hear well. So there are these fantastic hearing aids that are out there that enable people to hear. So if the damage is to the amplifier, then you can get a hearing aid. Unfortunately, sensory neural deafness stems from damage to the inner ear or auditory nerve. They are able now to have cochlear implants that can heal some of the or remedy some of that damage. But if the damage is done to the auditory nerve, unfortunately, there is no treatment as of yet. Maybe they'll come up with it, but there's no treatment for that. Sense of smell is the other one we have. Now, sense of smell and taste go hand in hand, right? If you can't smell something, you can't really taste it. The sense of smell is referred to as olfaction, please, or olfactory nerve. Please make sure you know that for the exam. It's detected through these chemicals that kind of evaporate and travel through air. So in the summertime, you can smell garbage worse than in the winter, or when a cookie is hot, you can smell it better. So, and what's fascinating, please make sure you know this for the exam. If you look right here, our sense of smell, the olfactory is right here, I guess, travel to the brain. Well, this area is the limbic system. This is your memory, emotions. This is all processed. So our sense of smell is closely attached to our memory and emotions. And sense of smell is the only one that doesn't have to travel through the thalamus to be processed. So if you've ever gone to, let's say, somewhere and you smell something and you, all of a sudden emotion comes through you, whether good or bad, you have a memory of that smell, um, emotional memory of that smell. So smell is really a strong trigger for our emotions and memory. We know smell also helps us contribute to the flavor of food. So quite often people smell something to know whether they like it or not. These odors are samples of molecules that are ultimately traveling through the air and trigger these receptor neurons in the olfactory membrane. Travels to the brain through the olfactory nerve. Last of our senses is a sense of taste. That's referred to as gustation. You don't need to know the five different flavors, but these are the 5 flavors that we have. Taste, we have these taste buds on our tongue. It used to be, you don't need to know anything more than just taste is gustation. If you ever get a chance, take a look at your tongue. It is an amazing muscle in our body, but it's really disgusting. We used to think that different areas of the tongue were responsible for processing different flavors, but we're finding that not to be true at all. We have these taste cells. These are receptor neurons on the taste bud that ultimately help us process the information, and they are found in the lower part, the side, and the uh, back of the tongue. What's amazing about the tongue, it is highly vascular. So if you have folks that pierce or have damaged the tongue, and they will bleed. But what's amazing about this structure is it heals itself really quickly. It is really absolutely. So if you ever get a chance, look at your tongue and look at the bottom of the tongue, and you'll see all the blood vessels that are there. Oh, sorry, I didn't want to shock you. So these are some of the conditions that are associated tongue issues. Apparently, this can happen with smoking where the cells die, and it's truly gross. So I'll end the class here.